How are humans able to detect taste? We'll start answering that question by looking at a tongue. Tongues are covered in taste buds, which each contain many different taste receptor cells. The big 5 taste receptors in humans are sweet, bitter, umami, sour, and salt. However, there are many more types of taste receptors like those for fats, carbonation, and water. We will begin by looking at a sweet-sensing cell on a taste bud, so let's zoom into its cell membrane. Sweet-sensing neurons have G-protein coupled receptor proteins TAS1R2 and TAS1R3. The sweet-sensing neuron only accepts sweet ligands to bind and cause conformational changes in the receptor complex. Once bound, the GPCR will activate the alpha, beta, and gamma subunits, and the G-protein complex will break apart from the receptor proteins. The G-protein subunits will migrate to a PLC beta-2 protein, activating it. Once activated, the PLC protein will migrate to a PIP2 complex, which will then dissociate into IP3 and DAG. IP3 is then able to migrate towards the cell's endoplasmic reticulum, where it binds to IP3R3 and changes the channel's permeability to calcium. Calcium will leave the endoplasmic reticulum and enter the rest of the intracellular space. Calcium will be able to bind to the TRPM5 ion channel, which increases the permeability of the cell to sodium ions. Sodium will rush into the cell and change membrane potential, resulting in an action potential if the threshold is reached. What about the other taste-sensing cells? Well, compared to the sweet-sensing neuron, the only difference from the umami-sensing neuron and umami taste pathway is that the TAS1R2 receptor is replaced with TAS1R1. This change results in umami ligands being necessary for conformational changes. With the bitter taste pathway, there's a different class of receptor present, T2R. With a T2R receptor, only bitter ligands are able to ignite the cascade. Lastly, what about sour and salt? Well, the pathways we have already seen have been G-protein coupled receptor pathways, where a ligand causes a conformational change and activates a G-protein cascade. These channels cause a relatively slow and indirect change in membrane permeability. Ion channel receptors, on the other hand, differ in that the ligand binds to an ion channel and opens it, allowing ions to flow through directly. Membrane permeability of the neuron changes almost instantaneously. These types of receptors are those involved in the sour and salt taste pathways.